Too. Well, I'm really pleased that Ben Prosser, who's really an expert on the mechanobiology of the nucleus, has agreed to talk with us. So I would not say that the order of the lectures this week follows the most logical pattern, but it's summer, people are on vacation, et cetera. But the goal is really by the end of the week for you to be able to put it all together. And one of the key parts of putting it all together is understanding how mechanics relate to the nucleus. CEMB is really, I think, one of the leaders, if not the leader, in understanding nuclear mechanics and nuclear mechanogenomics. And so Ben's going to give you an intro lecture because this is going to be important, I think, for you to think about no matter what approach and what project you're taking in your labs in the future. So thanks very much, Ben. And I would say the group, it's a little harder to ask questions when someone is online, but Ben is right here. And so do you mind if people?
No, I was actually going to say that's
Okay.
my first comments to please interrupt and raise your hand and ask questions during the talk. I'd rather not just talk at you guys for 45 minutes to an hour. It would be much more fun to be a conversation. If we don't get through my last 4 or 5 slides, it's not a big deal. Much more important to kind of have a dialogue. So if something is confusing and I don't know everybody's background, I'm sure people are of different backgrounds, different levels of expertise. So just interrupt as we go, all right? So as Becky alluded to, I'm tasked with sort of introducing you guys to the basics of nuclear mechanobiology. This image here is one of our favorite cell types in the lab, which is a cardiomyocyte or a heart muscle cell. And you can see these cytoskeletal elements in magenta and gold, the microtubules and intermediate filaments sort of impinging on this nucleus. And so one of the themes of today's talk is how are those forces that get transmitted to the nucleus sensed and what sort of biological consequences do they have? So our goals for today's lecture is to really try to appreciate how physical cues can play an equally important role as sort of biochemical cues that are much more well studied in regulating gene programs and thus cell development and behavior. And we want to gain a general sense of sort of where the field currently stands on mechanistic understanding of nuclear mechanotransduction. And if the term nuclear mechanotransduction feels vague now, hopefully it'll be more clear by the time we get to the end. And I really hope you guys will also be able to appreciate the requirement for using very cutting edge, sort of innovative tools and approaches in order to try to gain insight into nuclear mechanobiology. This is a challenging topic to study. So I'm sure you've heard these different sort of mechano terms. I feel like we put mechano and then anything at the end of it these days. And so I kind of wanted to get us on the same page in terms of the terminology, at least how I think about it. So mechanobiology, of course, sits at this interface of sort of biology and engineering. And it's the study of how physical forces and sort of changes in either cell or tissue mechanics contribute to either development, to physiology, or to pathology. And nuclear mechanobiology is just when the above involves the nucleus in some way. And of course, we'll talk about that in more details today. Now, mechanotransduction, this is the actual conversion of a physical force to a biochemical response. I would consider this akin to mechanosignaling. And so in the cellular context, this is typically where an extracellular force elicits some sort of intracellular signaling cascade and response. Now, what initiates that signaling cascade is the mechanosensor. So mechanosensing is a protein or a cellular structure that responds to that initial physical stimulus to initiate sort of mechanosignaling or mechanotransduction pathway. And one of the lessons I hope you guys will come away with from today's lecture is that the nucleus is an important mechanosensing organelle within the cell. Now, there are also, of course, different types of mechanical stimuli that a cell will sense. And so it's important to differentiate between them. So this is sort of our refresher on Mechanics 101. Hopefully these won't be new terms to you guys. We will be talking about stress on the cell, which can come in many forms, of course, defined by the force per cross-sectional area of the cell. And we can have that force could be a compression, which is a pushing force, tension, pulling force, bending, torsion, shear. Cells will experience all these different forces in different situations. Strain, we think about strain a lot in muscle. This is a unitless measurement. It's the change in length, delta L, over the initial length. So if you take a cell and you stretch it by 10 microns, and it's a 100-micron cell, that's about a 10% strain, right? It's a unitless measurement. Stiffness is the relationship between these two. Stress over strain, typically described in newtons per meter squared or in pascals. And the inverse of stiffness is compliance. So you'll often hear those terms used interchangeably. So this is sort of the typical stress-strain relationship for a cell. And so because stiffness is stress over strain, the slope of this relationship at any given point reflects the stiffness of that cell. And so you can see this sort of non-linear relationship between stress and strain in the cell. And so the cell's cytoskeleton is comprised of many different cross-linked sort of semi-flexible polymers. And as such, it exhibits this sort of very non-linear viscoelastic behavior that includes this power law of stiffening, getting increasingly stiff as you increase the stress on the system. All right, now I want to start by sort of showing some of the kind of prominent examples of nuclear mechanotransduction in action. And all cells are mechanosensitive, although for some it can play a larger role in their everyday biology than for others. And it'll probably become apparent by the end of this talk that we think a lot about muscle in my group, which is, I think, a really interesting cell to think about in terms of mechanotransduction. And so we can look at some of the common or widely studied examples of nuclear mechanobiology in development. I'll give an example in physiology and an example in disease. And I'm going to show these at first just at sort of the gross cellular or organ level, behavioral level. And the hope is by the end of the lecture, you'll be at least able to posit some of the sort of more molecular mechanisms that might be contributing to those sort of organ or cellular level responses. So first, a key role in development. And this is a rather famous example, perhaps one of the most famous examples of mechanotransduction that came right from a CEMB scientist at Penn, where Dennis Disher's group, now back in 2006, showed that the fate of a pluripotent stem cell can be decided simply by the stiffness of the substrate that that cell is plated on or cultured on. So here they took naive mesenchymal stem cells, and they plated them on either soft, on medium stiffness, or on stiff hydrogels or matrices, mimicking the stiffnesses that would be found in brain or in the muscle tissue or in bone. And interestingly, same cells on each of the different matrices, but only the stem cells that were plated on soft matrices developed these neuronal markers, where the ones plated on medium stiffness matrices developed these muscle markers, and those plated on the stiff substrate could differentiate into a bone-like cell. So I think this is really the first demonstration that the extracellular stiffness, sort of upstream of any other genetic or chemical cue, was sufficient to determine a cell fate choice. Right, and demonstrating really a vital role of mechanotransduction in development. And this paper was kind of a game changer for the field. I think it's been studied, or it's been cited like 15,000 times to date, which is a good number if you can get there. So, all right, so now a sort of a key role in physiological adaptation. And I like this one just because I used to be a runner anyway before my knees decided biking was more fun. So another common mechanotransduction mechanism is the sort of remodeling of bone or tissue that can happen in response to changing external forces. And I think this is a cool example, which is sort of the adaptation that can occur during barefoot running. And so if you look at impact forces, which is shown here on the left of either barefoot or shod runners, you see that shod runners, they tend to land first with their heels. And that heel strike results in this large impact transient that some have tried to link to running injuries. Whereas a barefoot runner, they tend to land more with a forefoot strike, which promotes this more decreased loading transient and more even distribution of force. Now, this doesn't mean, of course, you should just kick off your shoes, right, and run 5 miles barefoot. Most of these people that run barefoot have done so from a very early age. And as such, the bone, the musculature in their feet have considerably remodeled to support that force distribution. You guys are probably too young to remember this, but maybe 15 years ago, there was like a big barefoot running kick, and many people just threw off their shoes and ran out and started running on city streets. It didn't go well for many folks, right? But if you can allow that mechanobiology to remodel of that tissue, it can ultimately result in favorable adaptations. All right, and now another example from a key role in pathology. And so sort of being able to sense, being able to adapt to the external forces or the geometry of your environment is also a key a factor for cell motility. And this is an example from Jan Lammerding's lab, where they took breast cancer cells and they challenged them to migrate through this almost seemingly impossible narrow constrictions in this microfluidic devices. And the cells, you can see they not only they could make it through those very tight squeezes, which if you imagine, that has to be a somewhat Herculean effort to break down your cytoskeletal architecture and deform to the extent that they have to deform. But they could also then still generate enough sort of traction force to pull or push themselves through that tight space. This is an ability that cancer cells, you can imagine, are particularly good at and would favor their ability to metastasize or invade a tissue. But as you can see also from the spilling out of this GFP signal, which was localized to the nucleus, this tight constriction is actually inducing a rupture in the nucleus that from the cytoskeletal pressures that are on that nuclear membrane. But the cell could sense that, it could reform the nuclear integrity, and you actually see it pulls sort of the GFP back in, those spilled contents back in, and can carry on. And so I think this is a rather fascinating mechanism for an adaptation of a cell to be able to increase its invadability. Yeah, please.
I have a question. So what is a GFP tagged on, and
Yeah, it's
if it is being spilled out of the nucleus, so is there an like active mechanism that sort of tries?
pulls it back in. Yeah, so it has a nuclear localization signal on the GFP, so it will be sort of actively transported into the nucleus by default. And so the spilling out represents sort of a dissolution of the nuclear membrane into the contents, and then that NLS helps drive it back into the nucleus.
And during this process, the volume is still same. It's just the weight thing.
The volume of the nucleus or the
Yes.
volume? Well, that's a good question, whether water is squeezed out by the cytoskeletal pressures or not. I don't know if the volume of the nucleus would actually be, if you have a nuclear rupture, certainly the volume of the nucleus could change.
Right, like if it is kind of a drop, let's say, then at least from the images, like at least it's because it's on 2D, so one can presume that, well, it's not being, somehow, at least the volume is conserved in 2D. At least I can see that here. But
The area may be conserved, but if the nuclear pore ruptures and you lose molecules from the nucleus, right, that would create an osmotic gradient that could lead to also a loss of water and actually a reduction in nuclear volume. But I don't know if they measured that explicitly here.
Thank you.
Yeah, no, of course, great question.
Question. So
Yeah,
it looks like at the first, like discs, the nucleus kind of backs up and then projects itself forward.
Yeah.
But it doesn't do that for the second one. Do they have any explanation for that?
Yeah, that's a great question, and I don't think I know that biology well enough. I know there is actually quite a bit about sort of accumulation of cytoskeletal forces and sort of the rearward direction that can actually help propagate something through a tight constriction, and how that's going on here. What we're not visualizing here is the cytoskeletal harmonic that's also taking place during this process, and there's quite a bit of work about how that has to happen in order to generate the forces to push something through, and then whether that could be compromised when trying to navigate repeated constrictions. I'm not really familiar enough with that biology, but it's an interesting observation.
Thank you.
Yeah, of course. Okay, so I think that's a really remarkable feat when you try to think about it. And so how do cells sort of accomplish these amazing feats? And we can think about this in sort of the hierarchy of a typical mechanotransduction cascade. And again, this would often start with mechanosensing. So cells will be testing their environment, and mechanosensors use these sort of force-induced changes to transmit and convey information about the cellular environment. So you have adhesion receptors, other membrane proteins that can link the cell to its external environment, and other proteins and complexes link those mechanosensors then to the internal cytoskeleton. Now, once you've sensed that signal, it has to be propagated. And this can be either chemically or physically linked propagation. We'll talk about both of those mechanisms. So the extracellular matrix and sort of its interconnected network of cytoskeletal elements play really a central role of force conduits throughout tissues. Now that signal can be integrated at the nucleus, so either that chemical or physical signal can arrive at the nucleus, or you could have deformations that are transmitted to the nucleus, which can change its architecture, can change chromatin conformation and transcriptional patterns that can then modulate gene expression changes. And we'll talk in quite a bit of detail about that. And then finally, you have a cellular response. And you see this can bypass signal integration at the nucleus, right? You could have a mechanotransduction cascade that is completely independent of the nucleus, but it often involves the nucleus, and this can result in a wide range of responses, as you just saw in the previous examples. So the mechanotransduction response, it's often mediated by overlapping and often difficult to discriminate pathways. And some of the sort of most common mechanosensors and transducers are depicted here in a representative cell. And so I'll just point out a few. One, you can have these stretch activated channels or mechanosensitive ion channels that can be activated in response to stretching of the plasma membrane. This will allow the permeation of calcium or other ions that can act as a second messenger within the cell. Endothelial cells, for example, have a layer of what are called glycoproteins that are found on the surface of the cell called the glycocalyx. And these can sense this fluid shear stress in, for example, a vascular bed or stress that's applied parallel to the cell membrane, which can also initiate a mechanotransduction cascade. You have your cell-cell adhesions shown here, which include these adherence junctions and desmosomes that serve as sort of scaffolds that link cytoskeletons from connecting cells. And that can allow this sort of intercellular stress transmission between cells. And then you have sort of the classic cell and extracellular matrix interaction depicted down here, where the cell can sort of probe its external environment and sense changes in that extracellular milieu or the tissue strain that it then propagates through these fibrous networks. And that strain can be then propagated through the linked cytoskeleton, which hopefully you can see here, to, for example, the nucleus, which can sense and integrate those signals into some sort of cellular response. But this last part, sort of how force really modulates the transcriptional machinery, modulates gene expression, and how much of that is really due to force actually acting directly on the nucleus versus sort of a second messenger signaling cascade that reaches the nucleus. That has a lot of unknowns, and that's what we're going to focus on today. So this major missing link a lot is really how these signals are transduced to the nucleus, how that regulates gene expression programs. So we can think about this in sort of two broad mechanisms, and in terms of how force can regulate gene expression. And it's very difficult to untangle these two. The first would be sort of indirect nuclear mechanobiology. And so this is what we'll talk about in terms of force-dependent signal transduction. This is where you have more of a cytosolic signaling cascade, often initiated by forces at the cell periphery, say the activation of a mechanosensitive ion channel, that will ultimately result in, say, a transcription factor being imported into the nucleus and the activation of a gene expression program. The other is this more direct force transmission to the nucleus, which I find to be really fascinating. And this is where, a force typically transmitted via the cytoskeleton will physically push or pull on the nucleus to deform the nucleus and elicit some sort of biochemical change that can directly alter gene expression. These mechanisms, they're not mutually exclusive, and they likely each contribute, and perhaps synergistically, to the mechanoregulation of gene expression. And so one sort of demonstration of this interplay is in the development of the heart, or cardiomyogenesis. Yeah, please.
Can you tell us about the time scales at which how
We'll get exactly into that time scale, because I think it's a really important distinction in just a couple. Actually, I think it's the next slide. So, great question. Okay, so this cartoon is showing the sort of interplay of sort of indirect on the left and direct mechanotransduction on the right that underlies the specification and development of early cardiac cells. So on the left, you have this indirect force-dependent signal transduction. It starts with these integrin receptors, right, which can sense tissue stiffness through the extracellular matrix, but this activates a kinase signaling cascade that will ultimately result in some of these transcription factors or coactivators entering the nucleus and regulating gene expression. But the same tissue stiffness can also be sensed through integrin receptors, transmitted through the actin cytoskeleton to cause local deformations on the nucleus that affect the chromatin and which also can modulate a transcriptional response, or what might actually open the nuclear pore complexes to allow those transcription factors to more readily access the nucleus. So it's this latter component that we're going to talk about more, but that posits that there is a direct interaction between the cytoskeleton and the nucleus that can affect nuclear function. And it really posits the nucleus as a dynamic mechanosensor, right? Sensing that deformation and responding with some biochemical output. This is a relatively new field, I think, with a lot still to explore.
Question.
Yeah, please.
So I don't know much about it. I just know the name. So like Nesprin
Yeah.
is a protein that I think is.
Yeah, it would be like, it's not schematized here, but it would be right around here.
That's it. I just wanted to know where.
We'll get all into the Nesprin. So the Nesprin is one of the key components that helps link this cytoskeletal element to the outer nuclear membrane.
Thank you.
Yeah, of course. Yeah, please.
So yesterday
I'm
we,
so glad you guys are interrupting. Thank you. This makes it so much more fun for me.
so in the lecture yesterday, it was shown that focal activation kinases has connection with the F-actin and transmits the signals, but here is it that it's just not missing, or it's have two independent pathways, an indirect pathway and a direct pathway to how it does the signal transduction.
I'm sorry, maybe I missed the last
Fact.
part of the question. Yeah, fact, focal
That
adhesion
is
kinase.
listed as indirect. Yesterday we.
It can do a little bit of both, right? Because it can also
That's
influence
the-
cytoskeletal remodeling. So there's quite a bit of crosstalk between these two pathways. Yeah. It's just here to make the demonstration that the same mechanosensing structure can ultimately lead to both signal transduction cascades as well as direct deformation of the nucleus. But there's quite a bit of overlay. And again, this is why it's so difficult to really untangle the two as what is really driving the deterministic mechano response.
In terms of like the direct like nuclear deformation,
Yeah.
is there a way for you to be able to separate between like the chromatin remodeling and the pore opening? Like, are they like normally like separated if the actin does connect to the nucleus, or do they usually like go hand in hand?
It will depend on. It'll depend on a number of factors, and it's a great question. One will be location of where the site of deformation is, perhaps relative to the nuclear pore, how that affects the curvature of the nuclear pore, which can have a big effect on how the nuclear pore complex can open, as well as whether or not there are local chromatin associated domains at the membrane. So we'll talk about that as well. Chromatin is not evenly distributed along the outer nuclear membrane. So depending on where that cytoskeleton is connecting and the proximity of that force application to either the nuclear pore or to the underlying membrane and chromatin could convey some specificity in this response. But you guys are asking great questions about like, how do you get to specificity with sort of a force transduction signal cascade? And there's still a lot of unknowns with that.
Ben?
Yeah.
I just want to apologize. I have to leave and just say to the students, enjoy your labs this afternoon. They're both sort of wet, benchy labs, supposed to be labs.
Great.
Thanks a lot.
Okay, so in talking about the timescales question that we just heard, and so I do want to talk for just a moment about how that signal is sensed at the membrane, how it propagates to the nucleus. And so in response to an applied force, the cytoskeleton, it can also bend and buckle, and it can rapidly transfer that energy to other parts of the cell, such as the nucleus. And this rapid force transmission is really enabled and facilitated by having a very stiff cytoskeletal network relative to a soft cytosol. This will allow fast, long distance force transmission. So if you think about sort of a real world example of this, if you have a very tensed cable in air, and one person holding one end, one person holding the other, if you smack one end, you'll almost immediately feel that vibration on the other side, right? If you take a floppy rope and you put it in water and you slap one side, you're not going to feel any, it's going to be dampened by the time it reaches the other end. So this relatively stiff cytoskeleton relative to the cytosol allows for very fast and long distance force transmission in many cells. So, and we have considerable evidence that this can occur in cells. And so these are these sort of different just tensegrity models showing rapid force propagation to a nucleus in response to either shear or strain. And in terms of the time scales, this will allow deformation of the nucleus on a microsecond time scale relative to the typical milliseconds to second long timescales that you would need with some sort of linked biochemical signaling cascade, right? So if you're going for speed, if you're looking for rapid signal transduction of a mechanical signal, then this direct force transmission is the way to go. We can look at one relevant real world example of this in primary heart muscle cells. So this is looking at the microtubule network in a beating cardiomyocyte, right? Which is being electrically stimulated here at about 1 hertz, about the same rate at which your heart is pumping. And you can see these microtubules, they're sort of buckling under that compressive load every time. And that signal is propagated to the nucleus. So if you grab that heart muscle cell, which is shown in the transmitted light on the bottom, and you look at the microtubules on the top, you can see how they become sort of taut. And you can actually see the deformation of the nucleus there that's outlined by that microtubule cage. And sort of increasing the stiffness, the density of the microtubule network in heart failure is associated with augmented force transmission to the nucleus, which can actually have detrimental effects on both nuclear integrity and gene expression that can contribute to the pathology of heart failure when this sort of cross-linked cytoskeletal network is excessively stiffened. All right, so once that force is propagated to the nucleus, how does it actually regulate gene expression? And I want to talk about a few principles of genome organization, the regulation of gene expression, which you're going to hear much more about from Malike Lakadamiali tomorrow. So this is just sort of a teaser to that. But this is an electron micrograph of a nucleus. And you can see the two forms of chromatin, sort of heterochromatin, which tends to be more dominant at the periphery, and euchromatin within the majority of the nucleosol. You can also see what we were alluding to earlier, these very differential regions, like dense heterochromatin in some areas and not so much at others, right? Now, that heterochromatin, that is the compacted sort of closed form of chromatin that is associated with gene silencing. The activation of chromatin to its more open form allows gene expression to occur, and that is typically regulated by modification of these histones by different enzyme complexes that can either acetylate or phosphorylate these histone tails. Now, this can be reversed by different repressor complexes. This is sort of epigenetic regulation of chromatin that hopefully you guys are somewhat familiar with, that can lead to either gene silencing or activation. But it's important to recognize that genome organization, gene regulation, it's more complex than just euchromatin versus heterochromatin. The genome is organized into multiple higher order structures. So starting down here, we have the DNA, which is sort of tightly wound onto these nucleosomes to form chromatin. And these chromatin fibers can then form these interesting loops that can place certain regulatory regions, like as enhancers, right near others, such as promoters. These can then be further condensed into what are known as TADs, or topologically associated domains, which can be organized into sort of either active or inactive compartments. Now, some of these TADs can actually be physically associated with the nuclear lamina and nuclear membrane. And we're going to talk about these quite a bit today. This is known as LADs, or sorry about all this terminology, but we're lamina associated domains, which are typically associated with repression of associated chromatin. And these whole chromosomal territories, they can intermingle with one another. And so, for example, if you have a transcription factor that sits at the interface of these, it can activate several different genes that may be on very different chromosomes, right? Or very different regions in these sort of transcriptional factories. And so all this to say, when we think about nuclear mechanobiology, it's important to keep this in mind, as the mechanical forces of the cytoskeleton often will change nuclear shape or locally deform the nucleus. And you can think about the effect that that would have on the genome organized in that region and how that can regulate gene expression. So a lot of gene regulation is really about sort of the accessibility of these different genomic regions to activators of gene expression. And this can be shaped quite dramatically by mechanical cues. Yeah, please.
This actually might just be like, I just kind of forgot from like high school biology. So we should have like 23, right? Of like the different like colors associated, like different chromosomes.
How many different chromosomes we would have? Yeah, there's 2 copies of each chromosome and 20.
So it should be like, it should be like 23 like locations, right? Like how they're like color coordinated.
Are these, I'm not actually sure that they are color coded by the chromosome here.
Well,
Um,
what I mean is there should be 23 bundles, right? In our
Yes.
nucleus.
Whether it would be, it depends on whether the nucleus is diploid or haploid, right? So you can have different numbers of chromosomal copies. I mean, I think the point here is that like we tend to think of the genome, like when we map it out in a genome browser as somewhat linear, right? And so you have the chromosome 1 and then this region and this region. But the reality is in the cell, it's folded, it's organized in very complicated ways. And so the same transcription factor could activate a gene that's at position 1000, at the same time as activating a gene that's at position 400,000, right? Because of this sort of intermingling of these different sort of chromosomal territories. Now, the density inside the cell will depend on where that cell is in its cell cycle, in terms of DNA replication, whether it's polyploid, haploid.
Okay.
Sure.
Question that also kind of like basic high school biology. So if we visualize like our genes like this, like DNA along strings, do they ever get tangled? Like, is it possible for stuff to get tangled?
Yeah, it can be, and I mean, tangling almost implies to me an accidental process, but I mean, it is indeed, they can be tightly wound, which changes their accessibility to those things like transcription factors and polymerases and activators, and then they can be unwound to sort of facilitate that. Now, if you have sufficiently high concentrations of nucleotides, they can undergo sort of different processes like phase separation and et cetera, that can also make them inaccessible. So again, a lot about gene regulation is just about the accessibility of that chromatin or DNA region, to the different activators of gene expression. And that changes dynamically in response to many different cues and can be affected by local sort of high concentrations of chromatin that could lead to phase separation, which I think you could think of as sort of a tangled body that is then sequestered from the rest of the nucleosome.
Thank you.
And again, you guys will hear a lot more about this as well tomorrow. Malika should go into sort of a high level of detail about how this is regulated. All right, but I want to focus a bit more on these lamina-associated domains and the nuclear lamina. So again, the nuclear lamina is this, it's a meshwork of intermediate filaments. It lines the inner surface of the nuclear membrane and the nuclear envelope. It's made of these lamin proteins, predominantly lamin AC, as well as B1 and B2. And it really provides the primary structural support for the nucleus. So when you think about nuclear rigidity, it's primarily conveyed by the lamina and by chromatin. Those are the two major elements that influence the mechanics of the nucleus. Now, it's also a scaffold for a vast array of different proteins, transcription factors, and the interactions between these elements are highly sensitive to sort of the state and assembly of the nuclear lamina and its remodeling. And which chromatin regions are associated with the lamina can have big influences on gene expression in a cell. And so I want to walk you through one of my favorite papers that came out in Cell in 2017 from a group here, Raj Jain and John Epstein at Penn, where they looked at how LADs can again influence the differentiation of a stem cell into its appropriate fate. So in a pluripotent stem cell, which I think we have up here, where's the schematic? Up here, you have these sort of lineage specific genes. So say the genes that would encode for a cardiomyocyte, and they might be normally repressed in a pluripotent cell where the pluripotent genes would not be tightly associated with the lamina. They would be more accessible and more on, right? Then as that cell starts to commit down a progenitor route or towards a differentiated cell, such as a cardiomyocyte, you have this changed interaction where the lineage specific genes come off the lamina, where the pluripotent genes are moved to the lamina, where they are then repressed. So that cell will lose its pluripotency and it will start to commit down a particular fate line. And they can see this quite evident, quite elegantly by imaging for particular DNA loci in developing immature cardiomyocytes. So in the images here, in green is the lamina. In red is the loci for a particular gene called titin, which is a muscle-specific gigantic protein. It's actually the biggest protein in the human proteome. And what you see is that in the stem cell, It's very tightly associated with the lamina, and it's actually in this methylated region of chromatin that is typically repressed. But then as that cell matures or differentiates, you see that titin comes off of the lamina, is then present in the nucleosol, is accessible, is transcribed, and this helps to commit that cell towards a cardiomyocyte fate. Their demonstration of what happens when this goes wrong, if you muck with lamina-associated domains at the stem cell fate, you get what's called precocious differentiation, where those stem cells start turning into differentiated cell types too early in development and often going down an inappropriate fate path. So cardiomyocytes start expressing neuronal genes. They start having neuronal electrical activity, which is not good for your heart, right? So this is an important way that you can regulate cell commitment through regulating lamina-associated chromatin interactions. Please.
So like it's showing what you're saying on this, but like if the lineage specific genes are on the lamina, wouldn't that make them more susceptible to like the mechanics of the nucleus though?
Yeah. So that's one of the important consequences is whether part of what drives this lineage specific gene to come off is actually when these cells start to beat a little bit and you get mechanical forces exposed on the nucleus, which might cooperatively lead to more lineage specific activation of, say, cardiomyocyte genes. So it's interesting how the mechanical environment during development, and we know this in the heart, that the mechanical forces on the early heart are critical for its appropriate development. And this could be one mechanism that actually might facilitate that by almost knocking off those lineage specific genes if it could be properly focused. That's more theoretical, that's more hypothetical, I think, than proven. All right, so coming back to, we heard a question earlier about nesprins and how those forces from the cytoskeleton actually get transmitted to the nucleus and the nuclear lamina. So nesprins, which are these proteins that have this cache domain, which I don't think is schematized here, but here are the nesprins. Different isoforms. They sit on the outer nuclear membrane and they physically link the nucleus to these different cytoskeletal elements, whether actin, microtubules, or intermediate filaments. And those nesprins are in turn connected to the inner nuclear membrane through these SUN proteins. So nesprins and SUN are the two key components of the link complex. And those SUN proteins directly interact with the nuclear lamina and chromatin through a number of adapter proteins. So importantly, this really forms then a contiguous route, say from the extracellular matrix, through integrants, through the cytoskeleton, through the link complex to chromatin. So you have a direct route where your tissue stiffness in the ECM could then be transmitted all the way down to your DNA.
Could you go back a slide really?
Yeah, of course.
Could you explain so is Nesprin part of the link complex or is the link complex separate?
That's correct. Nesprin is part of the link complex. So link complex just stands for linkers of nucleo and cytoskeleton. And the two key protein components are the Nesprin proteins and the SUN proteins. And the specific interactions with different cytoskeletal elements comes from different isoforms of Nesprins. So you can see Nesprin 1 and 2, for example, are well known to interact with the microtubule network, where other isoforms like Nesprin 3 interacts preferentially with intermediate filaments. So the local expression of different Nesprin isoforms change the link complex and change the different cytoskeletal elements that are hooked up to the nucleus as one way to getting sort of preferential force transmission through different pathways. Make sense?
Yeah.
Okay. We're scheduled till 11:15, right? Yeah, okay, so I think I'm gonna skip this slide a little bit.
We technically have until 11:30 if people aren't too wiped out by the end also.
I don't want to wipe people. I feel like when you go longer than an hour, then you start losing the engagement. This is just to show that one of the points that I wanted to make is that there are typically forces from the cytoskeleton on the nucleus. These change a lot based on context, and a lot of particularly early mechanobiology was done in cells in 2D culture, often on stiff substrates. The forces experienced by those nuclei are probably quite different than cells in a 3-dimensional environment, okay? And so that's just one thing I want to sort of caution you guys to when you're thinking about the cells and systems that you're working on. Sometimes the 2D culture of cells on a glass substrate will have an extremely different force profile on the nucleus than a primary cell in the body. And we think about this a lot because, for example, we study cardiomyocytes. And one of the best mechanisms understood to regulate nuclear mechanobiology are these actin stress fibers that form over top of the nucleus and can flatten the nucleus in a 2D cell culture. They don't exist in heart muscle cells, right? And so the physical forces that a heart muscle cell is going to experience is going to be very different in situ than in a 2D culture. So that's just kind of more of a cautionary note.
So,
Yeah,
so to extend, you mean to say that like the cells can, obviously it's system specific, but the cells can like literally create certain structures that are completely absent and may not be even like natural when it comes to like 3D.
Yes, and I don't mean to say that actin stress fibers are irrelevant. They just might be, the cell will remodel its cytoskeleton based on its external environment, right? And so the environment that we place a 2D cell in culture, where there's literally nothing on top of it but air or solution and a very stiff substrate underneath of it, is not often replicated in tissue. Now, in certain tissues, in certain contexts, you may have something similar. But that but that's where we do most of our interrogation. So the cytoarchitecture that we study most commonly in 2D is often not represented in 3-dimensional tissues, which is just something to keep in mind. Doesn't mean that the lessons that we learn from that are irrelevant. They often point to pathways that are activated in other systems and in other contexts. But it's important to kind of understand how much the environment matters to cytoarchitecture, which will really change the mechanosensitivity of your system. Is that fair?
Yeah, and then also like I was just wondering, maybe it might not be a great question, but are there research done on like how they can locally apply forces at the nucleus, let's say at front end or the back end or like and elicit any response, because that would help us understand which part of the chromatin is kind of associated with region of the nucleus, right? I was wondering if are you familiar with that sort of like line of questions or like research that's.
I'm not sure that I understand the question entirely. Are you asking whether or not it's being well studied, whether local forces on say one side of the nucleus versus the other can lead to local deformations in rearrangements of chromatin
because
in that region?
people that you showed like they have like global, like you're applying force globally on the nucleus, right?
Yeah.
So I was wondering if localized forces can have
Yes, so the answer is yes, and I'll show you some direct evidence of that in the next couple slides. Now, how often that's happening in biology, a lot of what I'm going to show you guys today on nuclear mechanobiology is like proof of concept that shows like these mechanisms can exist, or you can see this sort of local regional control. The question of how that's happening during normal cell physiology is much more difficult to untangle, of course, because there's so many intermixed signals. Okay, so in thinking about these like actually supported mechanisms for force transmission to the nucleus, we can think about this in sort of 3 bins that I want to demonstrate. And one is the role of sort of these force-induced protein conformational changes, where force would lead to a change in protein secondary structure that may then allow for a new interaction or a new phosphorylation site that conveys some biological function. The other mechanism that we'll talk about is actual physical stretching of the nuclear envelope and how the downstream consequences of that. And the third is sort of direct forces influencing chromatin modification, either the opening or its compaction or its modification by epigenetic regulators. And we'll look at some of the best evidence that supports each of these mechanisms. But again, this is relatively new. It's an evolving field, I think with a lot still to learn. So in speaking directly maybe to that last question, one of the seminal papers that supported force-induced conformational changes locally on nuclear proteins, it really sought to answer this key question: is force on an isolated nucleus, take the rest of the cytosol out of the equation, is that sufficient to regulate gene expression? And so forget about focal adhesions, forget about transmission through the cytoskeleton. What happens if you just tug on the nuclear membrane? And this was done by Keith Burr's lab at UNC in 2016. So the way they did this is they applied force directly on isolated nuclei through the link complex component Nesperin-1. So they used magnetic tweezers to stimulate magnetic beads coated with an anti-Nesperin antibody, okay? And they measured the bead, these are the beads here. These have a Nesperin antibody on it, right? So they're locally hooking up to Nesperin at the link complex. And then they could apply a magnetic field to displace the bead. And if they displace the bead with a known force, the magnetic field, right, then through Hooke's law, F equals Kx, they can calculate the stiffness of the nucleus, right? Because they're displacing the bead with a known force. K is your stiffness constant. So from this, what they showed is that when they repeatedly stretch the bead or displace the bead, you see that the displacement is going down with repeated deformations or repeated applications of force. This means that the stiffness of the nucleus is going up, right? Because the same amount of force is causing it to displace less and less and less. What they went to show is that that force is actually leading to phosphorylation rapidly of this protein called emerin that is facilitating those lamin proteins to be recruited to the link complex through the sun protein. So in this Western blot here, you can see levels of lamin AC specifically, which is really what conveys the stiffness to the nucleus, that with repeated applications of force, you keep getting more and more of that lamin that's associated with the bead fraction, okay? They then went on to show that actually that also regulates gene expression changes and changes in cell aspect ratio. So really sort of a seminal paper to establish that the nucleus is a rapidly mechanoresponsive organelle independent of anything going on in the cytosol. Okay.
To clarify, so they used the bead and they were measuring the displacement, but then I'm confused on how the displacement is going down. What do you mean the displacement is going down?
So here you're measuring the total displacement of the bead with the same application of force over time. And you can see that the delta here, the difference is getting less each time that they subsequently add additional force application to the bead.
So, but the bead isn't being allowed to return to its original spots, or
This is the force application profile. So when they release the force, the bead will naturally come back to some extent, but it comes back to less and less of an extent. Again, suggesting that the stiffness of the system is going down.
Oh, okay.
And then they took that bead, just the bead complex, and probed to what's happening locally associated with that bead, and you get more of this lamin AC specifically, not more lamin B, but more of this lamin AC recruited there, which is locally stiffening the nucleus at the site of force application.
Could they have seen, so this is technically almost like a viscoelastic response though, right? Like if they had given that more time between each force pulse, do you think it would?
Yeah, they didn't really dive, you're right, you're absolutely right in thinking about that. And they didn't dive into the viscoelasticity or the stress relaxation components of this. You can already see some stress relaxation within the plots there. Okay. So this kind of sort of dynamic regulation of the lamina and the effect it has on gene expression is really a common theme of nuclear mechanobiology. And important work also helped to show that the nuclear stiffness is really set by this ratio of lamin A over lamin B. And interestingly, that this scales well with total tissue stiffness. So this is suggesting that sort of the nucleus is stiffening by regulating the ratio of lamin A over lamin B to match its surrounding external tissue environment across different tissues and different microelasticity scales. And so this role of the lamina in sort of stabilizing the nucleus, it's been reinforced in a number of elegant studies, including one here where they're actually looking at chymographs of the nuclear envelope. So you can look at the change in its structure as a function of time from top to bottom. And they looked at this on cells that were elongated on these sort of rectangular micropatterned islands relative to cells that are on little circular islands, which is denoted here. And they looked at the fluctuation of the nucleus, right? So the cells on the circular islands are in red, and you see these severe fluctuations, where the cells on the rectangular islands, which have higher levels of lamin AC in that microenvironment, have a more stable nucleus. They could then look at this if you depleted lamin and you put the cells in the rectangular items, they see these massive fluctuations, again, suggesting you've reduced nuclear stability. And if they overexpressed lamin when they were on the little circular islands, now they could stabilize the nucleus, really pinpointing a role of lamin AC in nuclear stabilization, and that this correlates with tissue stiffening. Please.
So if this correlates with tissue stiffness, do you know if there are any studies that are being done to look at how this affects tissue morphogenesis? Like this, could it affect the stiffness of the resulting tissue,
Yeah, well,
you know?
I don't know if this directly answers your question, but I mean, one of the really fascinating cases of this is with lamin mutations, which we probably aren't gonna have time to talk about at the end, but one of the interesting things is progerin, which is a mutation in lamin that leads to irregularities in nuclear stiffness. And it actually it's referred to as like the Benjamin Button disease because it refers to a very accelerated premature aging phenotype, which is interesting to think about when we talked about those lamin-associated domains regulating stem cell development and proper fate decisions. So indeed, if you muck with lamina stiffness, you will have consequences on tissue morphology, tissue development, and tissue stiffness, if that's answering your question.
Thank you.
Okay. Okay, so later the district group, along with others at Penn, including our lab, sort of helped look at the mechanism by which this tissue stiffness can drive stabilization of the lamina. And this is where sort of the force-induced conformational change of a nuclear protein came in. And so what they found that was really interesting is that when you have this increase in cell, and say tissue stiffness, extracellular matrix stiffness, that results in an increase in actomyosin stiffness and force transmission to the nucleus. And this deformation of the nucleus by the cytoskeleton, it actually leads to an obscuring of phosphorylation sites on lamin A. So you are now obscuring these phosphorylation sites, and that stabilizes the lamin A expression. If you soften the tissue, you soften the cytoskeleton, those phosphosites become exposed, and the phosphorylation of lamin A leads to its degradation. Okay, it becomes more soluble, it comes out of the lamina, and it's more readily degraded. So this was a mechanism for how tissue stiffening through actomyosin contractility can lead to a conformational change in lamin that makes it more or less susceptible to degradation, thus ultimately changing nuclear stiffness. Okay. So I want to touch on these two last mechanisms in our last 5 minutes. Getting away from the lamina for a moment. Another mechanism of nuclear mechanotransduction can involve stretching of the nuclear envelope. And this can have important consequences for the accessibility or the permeability of nuclear pores. So what the authors of this important paper are showing here on the top left is that when they give the cell an osmotic stress, either through hypoosmotic or hyperosmotic conditions, the cell will change in its morphology and the nucleus will change in its morphology. Something similar happens when you plate cells on stiff substrates. For example, here at 150, wow, that really doesn't respond well, sorry, kPa, the cells will flatten and the nucleus will flatten. But what they showed importantly is that when you stretch the nucleus, when the cell is flattened and the nucleus is flattened, that was increasing the nuclear pore size, and thus the permeability of nuclear pores to transcription factors, other small molecules that can transit that accessibility. And importantly, this led to the accumulation of YAP, which is this well-studied mechanosensitive regulator of gene expression. So the general idea, when you have a soft substrate, the cytoskeletal forces are not acting so much on the nucleus. You have a rounder nucleus. You have less opening of the nuclear pores, less YAP coming in. And then when you stretched that, or when you place that cell on a stiff substrate, the cytoskeleton deforms the nucleus, stretches the nuclear pores, you have more YAP translocation, and this turns on mechanosensitive gene expression. So this was one important potential mechanism for nuclear mechanotransduction, although it really raises this question again of specificity. Because here they're focusing on YAP coming in, but if you open nuclear pores, just by first principles, everything should be able to come in more readily. So how you achieve specificity of letting certain mechanosensitive transcription factors in and not others, I think really remains to be determined and is a question raised by this paper. All right, and for my last example, I want to talk about maybe the most direct mechanism of nuclear mechanotransduction, and that's chromatin stretching. And so the proof of concept for this was really provided in this rather beautiful paper using clever approaches back in 2016. And so what the authors wanted to test was whether directly stretching chromatin in itself was sufficient to turn on gene expression. And they did this in a very clever way. They engineered a artificial chromosome, or a bacterial artificial chromosome, that contained multiple copies of this gene, DHFR. Doesn't matter what the gene is, right? But it has multiple copies of this gene. And it also has multiple GFP tags or fluorescent tags on that artificial chromosome that are regularly spaced out. So you express that in cells, and in the nucleus, you see multiple little GFP puncta, right? Which is that artificial chromosome with these different tags on it. They then used a technique called magnetic twisting cytometry, which is where you take magnetic beads that are coated with a domain that hooks up to integrins, right, on that cell surface. You then use a magnetic force to twist the beads that will pull on the integrins and transmit that force through the cytoskeleton, through the link complex to the nucleus. Does that all make sense so far? Okay. So they did that, and then they asked what happens to those little GFP spots, right, which are sitting on chromatin when they apply this external force. And they measured it over time. And so here you're looking at the relative position of two of those GFP loci to each other when they apply this twisting force over time. And what you see is that as they twist, the spots get further away from each other, suggesting that you're stretching that chromatin, and then they come back close to each other as they complete or they reverse that magnetic twisting. So importantly, this was a demonstration that a force applied from the outside could lead to the stretching of chromatin. And if they disrupted the link complex, this wouldn't happen anymore. The same twisting force was not able to stretch chromatin, arguing that you really needed that cytoskeletal force transmitted through the nucleus to stretch chromatin. But the cooler thing that they did is also found that when they stretch, they rapidly induced the expression of this gene that they had engineered on this artificial construct, this DHFR. And that's kind of wild. Gene expression activation in 15 seconds after the application of an external stress. That's a lot faster than you'll probably ever see with some sort of biochemical signaling cascade. So again, while the sort of the physiologic, the pathologic significance of all this really remains to be further studied, this was a wild demonstration that forces applied on the outside of the cell can lead to seconds changes in gene expression through a direct physical force conduit involving cytoskeleton and the link complex to chromatin. Okay. So I think we're going to close there. I'm not going to tell you about where nuclear mechanobiology goes wrong and in laminopathy, other than to say that I think this is one of the really sort of fascinating now applications of this new understanding that we're getting of nuclear mechanobiology. So in patients that have mutations in lamin A, they primarily manifest as muscle diseases. Perhaps this is not surprising, as lamins are everywhere, but muscle is one of the most mechanically active tissues there are. And what we're learning now is that when you weaken the nuclear lamina, we already showed that that sort of destabilizes the nucleus. It renders it susceptible to these nuclear ruptures from cytoskeletal forces transmitted through the link complex, which leads to DNA damage. It leads to these wonky chromatin protrusions actually spilling out of the nucleus, which can lead to cell death. And so the understanding of that has actually led to the fact that we can now sort of break the link complex in these lamin deficient mice and dramatically increase their survival. So this is kind of wild, right? So in black and yellow are mice that have lamin depleted from them, and they die within 50 days or so, and typically of heart failure. However, if you, in those same mice, you actually delete the sun protein, one of those key components of the link complex, now those mice can live to a very much longer time, right? And so we can think about this, and this could have dramatic effects on patients where this is a rather devastating disease of muscle dysfunction and cardiac dysfunction and ultimately heart failure. So can you shield lamin deficient nucleus by breaking the link complex? And we followed up on this to show that actually the specific cytoskeletal element that you want to target is the microtubules that are interacting with nesprins to drive these excessive forces on lamin deficient nuclei. And that if you just take those same mice again, and you use a small molecule to disrupt the microtubules, where normally those mice will die within about 20 days of deleting lamin. This is looking at their cardiac function, their cardiac pumping. If you disrupt the microtubule network in those animals, you can preserve their cardiac function and expand their lifespan, which is opening up new avenues of treatment for patients with laminopathy. So I just wanted to share one application of sort of these principles of nuclear mechanobiology and how it might be manifesting its way towards clinical applications. All right, so with that, we can stop. I hope that I've showed you guys today about how mechanical transduction can underlie a number of fundamental processes and that the nucleus can be a direct mechanosensing organelle, and that force applied to the nucleus is sufficient to rapidly regulate gene expression, which can occur through multiple mechanisms. Although currently I think the role of sort of force-dependent remodeling of the lamina and downstream consequences is probably the best supported in terms of its biological importance. But again, a lot of this, as we talked about today, is open territory, still ripe for exploration. And hopefully you can appreciate that sort of the technical innovation that has come into to generating some of these insights, and that more will be required to really show its importance in physiology and pathology. You guys are a great audience. I really appreciate the questions. I'm happy to take any more, but this is the official end of the show.